Alright, hello everyone. I'm hitting you with another random science news stream. Got a few stories to go over today. I think it'll be pretty good. But let's jump right into the first one. This one, octopuses and squid octopi are masters of RNA editing while leaving DNA intact. So for those of you that don't know the way that our body works, let's pull it up. You can basically just do like central dogma of biology. With how we get our proteins and everything, we have our DNA, like our blueprint basically, and through transcription our body will take the information from the DNA and transcribe it into RNA, and then our body will translate that information from RNA to a protein. So basically this is saying that these, and through a few more, like, I guess, details, because I'm trying to think what it means by leaving DNA intact, like if it means telomeres, the ends of the chromosomes, or we'll see. Let's see. Grouse? Many writers grouse when an editor makes a change in a story, but the consequences of changing a single word usually aren't that dire. Not so with genetic instructions for main proteins. Even a small change can prevent a protein from doing its job properly. It's true. With possibly deadly consequences. Only occasionally is a change beneficial. It seems wisest to preserve genetic instructions as they are written, unless you're an octopus. Octopuses are like aliens living among us. They do a lot of things differently from land animals or even other sea creatures. Their flexible tentacles taste what they touch and have minds of their own. Octopuses' eyes are colorblind, but their skin can detect light on its own. Oh, I actually didn't know that. Hold on. Let me see if I can pull up that story too. I actually didn't know that. I want to read into that. They are masters of disguise, changing color and skin textures to blend into their surroundings or scare off rivals. To a greater extent than most creatures, octopuses squirt the molecular equivalent of red ink over their genetic instructions with astounding abandon, like a copy editor run amok. Very long-winded intro, but let's see. These edits modify RNA, the molecule used to translate information from the genetic blueprint stored in DNA, while leaving the DNA unaltered. Scientists don't yet know for sure why octopuses and other shellless cephalopods, including squid and cuttlefish, are such prolific editors. Researchers debating whether this form of genetic editing gave cephalopods an evolutionary leg or tentacle up, or whether the editing is just a sometimes useful accident. I mean, like most things, probably just an accident, probably just happened. But even some accidents could be beneficial. Scientists are also probing what consequences the RNA alterations may have under various conditions. So let's see, it's the... I'm getting lost in the metaphor and like what actually happens, I guess. Okay, squirt the molecular equivalent of red ink over their... Okay, so they squirt something over their DNA or whatever, I guess, is what it's getting at. Let's see. Some evidence suggests editing may give cephalopods some of their smarts, but could come at the cost of holding back evolution in their DNA. Interesting. These animals are just magical, says Caroline Albertin, a comparative developmental biologist at the Marine Biological Laboratory in Woods Hole, Massachusetts. They have all sorts of different solutions to living in the world they come from. RNA editing may help give the creatures vast numbers of solutions for problems they may face. I guess here's a video of how an octopus crawls. Which is interesting. I also never thought of like how an octopus crawls necessarily. Let's see, how cephalopods modify their RNA. Molecular biology, central dogma, yep, there it is, holds out instructions for building an organism that contain DNA. Cells copy these instructions into messenger RNAs or mRNAs. Then cellular machinery, called ribosomes, read the mRNAs to build proteins by stringing amino acids together. Most of the time, the protein's composition conforms to DNA template for the protein sequence of amino acids. And kind of like I said earlier, if any bit of this information is like... So our DNA is something called degenerative, which means that even if there's a few slight changes, the body might be able to make up for it because it might read it the exact same way. But if there's a change that it can't read said amino acid or whatever the same way, then you're definitely going to get a different amino acid in your protein, and then as a result, your protein could have a widely different structure to it. So it could just not work altogether. Let's see, but RNA editing can cause divergences from DNA instructions, creating some proteins that have different amino acids than specified by the DNA. Okay, I go into something that's going to go into it. Editing chemically modifies one of RNA's four building blocks or bases. Those bases are often referred to by the first letters of their names, A, C, G, and U, for adenine, cytosine, guanine, and uracil. I was literally about to explain those, but again, I was just like, it goes over it. RNA's version of the DNA-based thymine. Yeah, so your DNA will have A, C, G, and T bases, and RNA will have A, C, G, and U, which I'm trying to remember. Let's see. Because uracil and uracil is basically thymine, but it's got this extra little. Thymine has this extra little methyl group on the side here. Yeah. And so basically with RNA, it doesn't have that as uracil instead, which I actually don't. I'm actually not sure why that is. Let me see. I wonder why RNA would have maybe it's like the structure of the sugar backbone or something. Let's see, why does RNA have uracil instead of thymine? Instability doesn't matter as much for RNA. Thymine is also quickly oxidized. Okay. So, yeah, just basically works out better. Alright, let's see. There are many ways to edit RNA letters. Cephalopods excel at a type of editing known as adenosine to inosine, or A to I editing. This happens when an enzyme called ADAR2 strips the nitrogen and two hydrogen atoms off the adenosine. That chemical peel turns adenosine to inosine. So let's see. Inosine looks like... Is that inosine? Or is that a different one? Looks different. Okay, so here's basically the adenine, and here's the inosine that results. It's changing this group to oxygen. This double bond gets broken. That nitrogen gets a hydrogen on top of it. Okay. So what does that do for them? Ribosomes read inosine as guanine instead of adenine. Interesting. Sometimes that switch has no effect on the resulting protein's chain of amino acids. Again, because DNA is degenerative. Sometimes the changes don't have large effects. But in some cases, having a G where an A should be results in a different amino acid being inserted into the protein. Such protein-altering RNA editing is also called RNA Recoding. Sulfide cephalopods have embraced RNA recoding with all their arms, while even closely related species are more tentative about accepting rewrites, Albertson says. Other mollusks don't seem to do it to the same extent. I wonder if they're going to go into the... Well, I guess they said that they have this enzyme in particular, but is that the only difference? Only reason they do it? RNA editing isn't limited to creatures of the deep. Almost every multicellular organism has one or more RNA editing enzymes called ADAR enzymes. Okay. Short for adenosine deaminase that acts on RNA, says Joshua Rosenthal, a molecular neurobiologist, also at the Marine Biological Laboratory. Cephalopods have 2 ADAR enzymes. Humans have versions of them too. In our brains, we edit a ton of RNA. We do it a lot, Rosenthal says. Over the last decade, scientists have discovered millions of places in human RNAs where editing occurs. But those edits rarely change the amino acids in protein. For instance, Ellie Eisenberg of Tel Aviv University and colleagues identified more than 4.6 million editing sites in human RNAs. And even that many, I'm sure there's like a lot more that we gotta find. Of those, only 1517 recode proteins. Researchers reported last year in Nature Communications. Oh, recent. Of those recoding sites, up to 835 are shared with other mammals, suggesting that evolutionary forces preserve editing at these locations. Here's the... Okay, so here's how the RNA editing works. I guess from the adenosine to the inosine. Again, I try to go over something and it's just like, okay, it's in there. Cephalopods take RNA recoding to a whole new level, Albertson says. Longfin squid have 57,000 recoding sites. Damn. And we have how many? Okay. 4.6 million editing sites, 1517 recode proteins. Rosenthal, Eisenberg, and colleagues reported in 2015 in eLife. Since then, the researchers have examined multiple species of octopus, squid, and cuttlefish, each time finding tens of thousands of recoding sites. Soft-boiled or coleoid cephalopods may have more opportunities for editing than any other animal because of where at least one of the ADAR enzymes, ADR2 specifically, I guess, is located in the cell. Most animals edit RNAs in the nucleus, the compartment where DNA is stored and copied into RNA. Yep, that makes sense. Because if you're usually once you're trying to edit things or do whatever, I guess outside the nucleus with DNA or RNA, it might like break apart or it's already on its way to its destination, you know. So let's see, but cephalopods also have the enzymes in the cytoplasm, the cell's jelly-like guts. Plus it's like, with the, you know, in theory, with the enzymes outside of the nucleus, like once the RNA is going out to be translated into a protein, well, it can go under a whole new level of editing, which, granted, there are like tons and tons and tons of levels of editing for your DNA information in our bodies, but that's pretty interesting that it can happen at, I guess, any step of the way for the RNA. So let's see, having editing enzymes in 2 locales doesn't fully explain why cephalopods' RNA recoding so far outstrips that of human and other animals, nor does it explain the patterns of editing scientists have uncovered. RNA editing may give cephalopods flexibility. Editing isn't an all-or-nothing proposition. Rarely are all copies of an RNA in a cell edited. It's much more common for some percentage of RNAs to be edited while the rest retain their original information. The percentage or frequency of editing can vary widely from RNA to RNA, or between cells or tissues, and may depend on water temperature or other conditions. In longfin squid, most RNA editing sites were edited 2% or less of the time. Albertson and colleagues report last year in Nature Communications. But their researchers also found more than 205,000 sites that were edited 25% of the time or more. Yep, everything in your body is very variable and very specific to its situation. In most of a cephalopod's body, RNA editing doesn't often affect the makeup of proteins, but in the nervous system it's a different story. In longfin squid's nervous system, 70% of edits in protein-producing RNAs recode proteins, and RNAs in the nervous system of the California two-spot octopus are recorded 3 to 6 times as often as in other organs or tissues. Some mRNAs have multiple edit sites that alter amino acids in the proteins the mRNAs encode. In the longfin squid's nervous system, for instance, 27% of mRNAs have 3 or more recoding sites. Some contain 10 or more such sites. Combinations of those editing sites could result in multiple versions of a protein being made in a cell. There's also another process. Oh gosh. Let's see if I can find it. If I can... God, I can't believe I forgot the term. But there's an extremely specific term for how your RNA will be edited. Wish I could remember this. See, this is my problem with vocabulary, is a lot of it will just get lost for me. Splicing. That's what I'm thinking of. Yes, okay. Okay, I was about to be like, well, it's a tiny picture, that doesn't help. So your DNA can look like this. And, you know, the bases that are actually going to be used for a protein will be in the exon regions. And any bases that are just relatively, for lack of a better term, extra, and won't be translated into a protein or introns. And so basically, I guess this doesn't give like the greatest variability visual of it, but so anyways, like, you know, you have your pre-mRNA, then you take out the introns, then you slap all the exons together, then you have your mature RNA. But what this doesn't show is, you know, you have your exon 1, 2, 3, 4, but the RNA could create a different protein altogether by doing, say, like, it might not use 3 for this specific protein. It might be like 1, 2, 4, or like just 1 and 4, or maybe 2 and 3. So our bodies can also have different, you know, multiple proteins can actually come out of the same gene, just depending on what kind of editing needs to happen or what kind of splicing needs to happen. So it's pretty interesting how that works. All right, let's see. I'm sure they'll probably get into it in like 2 seconds. So having a wide selection of proteins may give cephalopods more flexibility in responding to the environment, Albertson says, or give you a variety of solutions to the problem in front of you. In the nervous system, RNA editing might contribute to flexibility in thinking, which could help explain why octopuses can unlock cages or use tools, some researchers think. Editing could be an easy way to create one or more versions of a protein in the nervous system and different ones in the rest of the body, Albertson says. When humans and other vertebrates have different versions of protein, often comes from having multiple copies of a gene. Doubling, tripling, or quadrupling copies of a gene results in a whole genetic playground to allow genes to go off and do different functions. And the genetic playground the DNA is. The cephalopods tend not to duplicate genes, instead their innovations come from editing. Okay. So editing is maybe more to make up for them not duplicating genes as much. And there's a lot of room for innovation. In squid, mRNAs for building the alpha spectrin protein have 242 recoding sites. All the combinations of edited and unedited sites theoretically could create up to 7 × 10 to the 72nd forms of the protein. Rosenthal and Eisenberg report in this year's issue of Annual Review of Animal Biosciences. To put this number in perspective, the researchers wrote, suffice it to say that it dwarfs the number of all alpha spectrin molecules. For that matter, all protein molecules synthesized in all cells of all the squids that have ever lived on our planet since the dawn of time. Damn. That incredible level of complexity would be possible only if every site were independent, says Kavya Rangan, a molecular biologist at the University of California, San Diego. Rangan has been studying RNA recoding in California's market squid and longfin squid. Water temperature triggers the squid to recode motor proteins called kinesins that move cargo inside cells. In longfin squid, the mRNA that produces kinesin 1 has 14 recoding sites, Rangan has found. She examined mRNAs from the optic lobe, the part of the brain that processes visual information, and from the stellate ganglion, a collection of nerves involved in generating the muscle contractions that produce jets of water to propel the squid. So wait, let's see. Same mRNA from optic lobe. Oh, interesting. Okay. I didn't. Again, I'm not exactly the most knowledgeable on marine biology, but I didn't know that there was a specific, I guess, term for the nerves that are responsible for the muscle contraction to help propel squids. Each tissue made several versions of the protein, but certain sites tended to be edited together. Rangan, Samara, Peterson, also of UC San Diego, reported last September in a preprint posted online at bioarchive.org. Their data is suggesting that editing of some sites is coordinated and very strongly rejects the idea that editing is independent. Rangan says, the frequency of the combos that we see don't match if every site was edited independently. Yoking editing sites may prevent squid and our cephalopods from reaching the pinnacles of complexity that they're theoretically capable of. Still, RNA editing provides cephalopods a way to try out many versions of a protein without getting locked into a permanent change in DNA, Rangan says. That lack of commitment puzzles Jianxi Shang, an evolutionary geneticist at the University of Michigan, Ann Arbor. This doesn't make sense to me, he says. If you want a particular amino acid in a protein, you should change the DNA. Why do you change the RNA? That's a good question. Is there evolutionary value to RNA editing? Perhaps RNA editing provides some evolutionary advantage. To test the idea, Sheng and then graduate student Daoan compared synonymous sites where edits do not change amino acids, with non-synonymous sites where recording happens. Okay, yeah, so synonymous mutations, you could say, they basically don't create any sort of change in the protein that comes out as a result. Hello, Rango. Welcome, welcome. And then non-synonymous is basically any other mutation that would create a difference in the protein. Supermeth? It depends what you would want to label as supermeth. Because honestly, I feel like if the scientists truly wanted to, then maybe. But there are probably some labs where they're studying it, but they got some really, really pure meth. And I know I worked in a lab with a lot of really pure cocaine. I say a lot, it was like a small vial. It became a pain in the ass to get when the Evergreen had its little oopsie daisy. So let's see. Since synonymous edits don't change amino acids, the researchers considered these edits to be neutral as far as evolution is concerned. True, but there can still be like a few effects from a protein with synonymous edits. It's alright, I'll forgive you for it, but I can understand the sentiment of creating a crime against nature. In humans, recoding or non-synonymous editing happens at fewer sites than synonymous editing, and the percentage of RNA molecules that are edited is lower than at synonymous sites. If we assume synonymous editing is just like noise that happens in the cell, and non-synonymous editing is less frequent and at a lower level, this suggests non-synonymous editing is actually harmful, Shang says. Even though recoding in cephalopods happens much more frequently than for humans in most cases, recoding is not advantageous or adaptive for cephalopods. This basically is saying that cephalopods can edit their, they can edit their RNA and DNA, or well, their RNA much more frequently than we can to create changes in their proteins. And these scientists are saying that basically it's not really providing anything good, like anything extra for them to be able to have that extra step of RNA editing. Let's see, there are a few shared sites for octopuses. Again, is it octopuses or octopi? This has happened too many times now, it bothers me. Wait, let's say, are octopedes? What? Oh, octopodes. What the hell is this? Octopus and octopi are acceptable plurals for octopus. Damn you, English language. The proposed plural octopodes is based on the plural of the ancient Greek word from which octopus ultimately derives. Interesting, I've never heard octopode before. Okay. So let's see. You shared sites where octopus, squid, and cuttlefish all recode their RNAs. The researchers found, suggesting that recoding is useful in those instances. Okay, so maybe a slight advantage, but very specific. This is a small fraction of editing sites. A few other sites are edited in one species of cephalopod, but not others were also adaptive, Zhang and Jing found. If it's not all that helpful, why have cephalopods persisted with RNA recoding for hundreds of millions of years? Because, to be honest, it's not really a thing they can change themselves. RNA editing may stick around not because it is adaptive, but because it's addictive, Shang says. Okay. Explain. He and Jing propose a harm-permitting model that is a situation that permits harmful changes to DNA. Okay, so I guess just some sort of like, yeah, some sort of radiation or whatever. Imagine, he says, a situation in which a G in an organism's DNA gets mutated into an A, okay? If that mutation leads to a harmful amino acid change in a protein, natural selection should weed out individuals that carry that mutation. Maybe, but you know, if, it depends on the level of harm that changing this amino acid would, or changing this base would give. If it's like a developmental thing, like, and that's the thing, you think that any like harmful change would weed out individuals via natural selection or whatever. But I mean, you know, classic Jurassic Park quote, life finds a way somehow. Let's see, so if they by chance the organism has RNA editing, the mistake in the DNA might be corrected by editing RNA, essentially changing the A back to a G. Oh, this is true. Yeah, so no matter how fucked up your DNA can get, you can just always fix it with the RNA level. If the protein is essential for life, then the RNA would have to be edited at high levels so that nearly every copy is corrected. When that happens, you're locked into the system, Shang says, now that the organism is dependent on RNA editing machinery. It cannot be lost because you'll require the A to be edited back to G for survival, so the editing will be kept at high levels. In the beginning you really didn't need it, but after you got it, you became addicted. Okay. So basically, if there's a DNA mutation, the RNA editing can fix it, but as you can imagine, you're gonna have to keep doing that for every single time you try to get that protein from the DNA. So your body, your cells will just keep going and going and going. Zhang argues that sort of editing is neutral, not adaptive, but other research suggests RNA editing can be adaptive. Welcome back. Alright, lurk away. I'll probably go through like 5 or 6 more stories after this. Let's see, RNA editing may work as a transition phase, letting organisms try out a switch from adenine to guanine without making a permanent change in their DNA. Over the course of evolution, sites where adenines are recorded in RNA in one cephalopod species are more likely than unedited adenines to be replaced with guanine in the DNA of one or more related species. Okay, that was a long sentence. I'm gonna need to reread that. Okay. Try out a switch without making a permanent change in your DNA. That would be interesting. If that was something they could actually control, that would imply that it's just like, oh, you can edit your RNA and be like, you know, does this help me? Is this harmful? You know, see what works. I wonder at all if this kind of thing could be done in humans to fix any kind of really bad mutation in DNA or something. That would be interesting. Okay, let's see. So let's see, over the course of evolution, sites where adenines are recorded in RNA in one cephalopod species are more likely than unedited adenines. Okay, so adenine and RNA. And, okay, whatever. And for heavily edited sites, evolution across cephalopods seems to favor a transition from A to G in DNA, rather than cytosine or thymine, the other two DNA building blocks. That favors the idea that editing can be adaptive. I mean, theoretically, you could argue it that really any evolutionary, anything that you find in an animal or something, can be adaptive, just due to like whatever specific situation that you could find it being advantageous in. Other recent work by Rosenthal and colleagues, which examined A to G replacements in different species, suggests that having an edible A is an evolutionary boon over an uneditable A or a hardwired G. Huh. Here's what I was talking about earlier with the RNA recoding. It's basically all those cephalopods have shitload of editing sites, and you know, all these other species having significantly less. Let's see, there's a lot of open questions. Evidence for and against RNA recoding's evolutionary value has come mainly from examining the total genetic makeup or genomes of various cephalopod species. The scientists would like to directly test whether recoded RNAs have an effect on cephalopod biology. Doing that will require some new tools and creative thinking. And since this is relatively recent study, it'll probably be a couple of years looking into that. Also curious, I mean, you know, just looked at cephalopods here. I guess they have zebrafish, but I wonder if that can be something in some other, like, marine life species. I'm gonna skip this one. Rather it just be background. So let's see. In fact, we're gonna turn it down just the tiniest bit. Okay. So let's see. Rankin tested synthetic versions of squid motor proteins and found that two edited versions that squid make in the cold move slower but traveled farther along protein tracks called microtubules than unedited proteins did. Okay, so basically made some synthetic versions. And the unedited, or the edited microtubules, or no. The edited motor proteins traveled further along the microtubules than the unedited ones. But that's in artificial laboratory conditions on microscope slides. True. Does have to be put to the test a little bit harder. To understand what is happening in cells, Rankin says she would like to be able to grow squid cells in lab dishes. Right now, she has to take tissues directly from the squid and only get snapshots of what is happening. Lab-grown cells might allow her to follow what happens over time. And with how lab-grown cells are, whenever we do human cells, we usually use something like an immortal cell line. And so it's usually like some type of cancer, which just allows it to grow and grow forever, so you can just essentially keep growing it, and you'll never have to worry about it getting old. Arguably. But I guess for squid, we don't exactly have, as far as I'm aware, an immortal cell line for those kinds of cell studies. So yeah, you would just have to keep either have squids reproducing so that you could get either like, because I don't know if they're doing this in young squids or in adults, but yeah, you would have to keep getting those squids from somewhere, and I don't know the, I guess, availability of them. Squidward. Yeah, you know, I will say, I don't know squids fuck. I don't know how good their reproduction is. And by fuck, I mean like fuck crazy, fuck wild. Or if squids have a relatively, you know, just have like, say, a couple of offspring. Yeah, so if it is slow reproduction, then that can also be a problem with studying it. But let's see. Zhang says he's testing his harm-permitting hypothesis by getting yeast hooked on RNA editing. Baker's yeast doesn't have ADAR enzymes, but Zhang engineers trained the yeast to carry human version of the enzyme. Okay, yeah. The ADAR enzymes make the yeast sick and grow slowly, he says. To speed up the experiment, the strain he is using has a higher than normal mutation rate. So this is another thing with cell studies, is you always have to make up for, like, so say you do use an immortal cell line, like a cancer cell line, you then have to keep in mind, like, okay, well, clearly there's mutations in the cell line. Does that have any effect with what I'm trying to do? I'm surprised you're going to a server with only 5 people. I guess it's not super popping right now. Alright, I think we're almost through this one. So yeah, so due to putting in this enzyme, then making the yeast slower needs to have a higher than normal mutation rate for this yeast. Let's see, it may build up G to A mutations. A perk that makes your ass clap very interesting. But if RNA editing can correct those mutations, the ADAR carrying yeast may grow better than the ones that don't have the enzyme. And after many generations, yeast may become addicted to editing, Zhang predicts. I don't know if I super like the term addicted in this sense, but I think I understand what they're saying. Albertin, Rosenthal, and colleagues have developed ways to change the genes of squid with the gene editor CRISPR-Cas9, classic. The team created an albino squid by using CRISPR-Cas9 to knock out or disable a gene that produces pigment. Researchers may be able to change editing sites in DNA or in RNA and test their function, Albertin says. The science is still in its early stages and the story may lead somewhere unexpected. Still, with cephalopod's skillful editing, it's bound to be a good read. Well, if we remember it in, say, a year or two, I guess we'll check up on the cephalopods and the teams and see how they're doing. Oh yeah, that's right. I wanted to read into this real quick, because the other article was saying that octopi can actually see sunlight through their skin, and I didn't know that. Little do people know that does happen in labs all the time. We are all screaming. Let's see. Octopus skin can detect light and respond to it. No eyes or brain required. No brain required. Tests of French skin samples from California 2-spot octopuses show stability clearly for the first time in any cephalopod, says Todd Oakley of the University of California, Santa Barbara. White or blue light prompts the pale skin's tiny, quick-change color organs, or chromatophores, to expand, creating waves of yellows and browns. The octopus tests, along with another research team's new studies of 2 kinds of cuttlefish and squid, feed discussion about whether light detection in places other than eyes plays some role in cephalopods' changing color displays. Maybe. All 4 species studied have light-sensing compounds in tissues beyond their eyes, the 2 teams reported May 15th in the Journal of Experimental Biology. Who knows? Biologists have known that eyes and the central nervous system have a major influence on prompting color displays that camouflage octopuses or let them communicate. A paper in 1993 foreshadowed this, reporting that octopus skin itself also seemed to respond to light, Oakley says. His co-author and UCSB colleague, Desmond Ramirez, worked for months determining that light triggers a color change displayed in detached octopus skin samples. Hold on, let me... Okay. We'll update the title every change in article. Alright. Eye's main light-sensing protein, one of many forms of opsin, may have gained another function in skin as a sensor for light, the researchers speculate. The idea fits with Ramirez's discovery that blue-green light prompts the quickest start for the skin sample's color changes. Light isn't very different, just 10 nanometers long in wavelength from the blue light, and most strongly simulates the eye's opsin. Genes known to encode compounds that work with opsin also turn on the octopus skin, the experiments show. Oh, sorry, what was opsin again? Main light sensing protein. Okay, light sensing protein. Gotcha. Early days, yeah. Usually most science is. Let's see, opsins and compounds that work with the proteins also show up in skin from the common and broad club cuttlefishes and the longfin inshore squid. The combination of compounds represents the first molecular evidence that opsins are functioning in the skin of these species. Also, molecular evidence suggests a location in the skin, the chromatophores. Makes sense. So far, though, there's no evidence in squids and cuttlefishes that light-striking skin is enough to make chromatophores blush. That really is what we're trying to figure out, is, like, can fish blush if we shoot them with the right amount of light? Cronin isn't completely ruling out the idea yet, and speculates about more subtle roles. Maybe they don't respond quickly, or directly, but they may alter a signal sent from the central nervous system, he says. Research on light detection beyond eyes and brains has been neglected for some time, says developmental biologist Florian Gravel. Yet, non-eye light sensing structures or compounds show up in the tube feet of sea urchins and the body walls of fruit fly larvae. Oh, I didn't know that. Polychaete worm flees light even after beheading. Interesting. Oh, I wonder what studies are being here that they were like let's figure out what happens to this worm after we cut its head off and see if its body can still sense the light. Okay, so this is the octopus, I guess. Okay. It's semi-hard to tell, but I guess it is changing color, but I don't know how quickly an octopus can change color, I guess. But it seems relatively slow after, like, it's not an instant, like, light turns on, boom, you know? Like, skin change. That was a sudden end to that music. Seen through a microscope, first at natural speed. Wait, actually. Okay, I guess. This internet's just being weird. Let's see. Seen through a microscope, bit of skin removed from an octopus does not react to red light, but when light shines on it, dot light color, change organs, expand, turn yellow or brown. Researchers call this process LACE for light-activated chromatophore expansion. Alright, so that was a little addendum onto the last thing. What do we got now? Oh yeah! So this one's about lucid dreaming and what it might tell us about our sleeping minds. So let me change that real quick. Oh. Which I was learning something the other day. If anyone's heard about, I was learning about reality shifting, because I saw a post about it recently, and I knew about the whole, like, craze of it and everything, like, a couple of years ago, but I was trying to read a bit more into it, being like, what? Like, I was like, who exactly, like, started this reality shifting craze and everything? And the more I read into it, the more I was just like, oh. I was like, so this is all very, like, close to lucid dreaming. But they're like, no, we swear it's not lucid dreaming, though. Like, it's actually shifting realities. And, you know, me as a scientist, I'm like, if you show me a study saying that somehow this is in your mind to another reality, then sure. But it sounds like you're lucid dreaming, and people have lost a little bit of their minds. Anyways, let's see what lucid dream can tell us about our sleeping minds. Alright, let's see. When Christopher Mazurek realizes he's dreaming, it's always the small stuff that tips him off. The first time it happened, Mazurek was a freshman at Northwestern University in Evanston, Illinois. In the dream, he found himself in a campus dining hall. It was winter, but Mazurek wasn't wearing his favorite coat. I realized that, okay, if I don't have the coat, I must be dreaming. That was just me putting in my headphones, don't worry. Mazurek says, that epiphany rocked the dream like an earthquake. Gravity shifted and I was flung down a hallway that seemed to go on for miles, he says. My left arm disappeared and then I woke up. I'm glad he kept those random details of his dream in there. Hey, you're doing science. Let's see, most people rarely, if ever, realize that they're dreaming while it's happening, what's known as lucid dreaming. But some enthusiasts have cultivated techniques to become self-aware in their sleep and even wrest some control over their dream selves and settings. Mazurek, 24, says he's gotten better at molding his lucid dreams since his first whirlwind experience, sometimes saying them as opportunities to try flying or say hi to deceased family members. Damn. I think most people that realize they're dreaming, I think I've only lucid dreamed for say more than the initial 10-30 seconds once or twice, but every other time it's I'm like, oh, I'm dreaming, and then it's my body's then you're waking up right now. They're like, we're not letting you do anything. And then even sometimes I'll realize I'm dreaming. I'll be in the dream, I'll be this doesn't make any sense. But I'm still just but let's go with it. Let's see where this goes. Or in the dream, I'll be oh, this doesn't make sense. I have to do this to wake up. And then I start doing some nonsense. Other lucid dreamers have used their personal virtual realities to plumb their subconscious minds for insights or feast on junk food without real-world consequences. But now scientists have a new job for lucid dreamers, to explore their dreamscapes and report out in real time. How messy are labs in real life normally? They can get pretty bad, honestly. It's essentially the same as just any other, say, average bedroom. We do usually clean up before inspections, though. Make sure it's nice and tidy. But in my experience, you usually have your area with all the chemicals and things. And then there'll be long work benches as well with any chemicals on shelves and whatnot. Because, it just depends on if the person individually is messy or not. Sometimes it can get annoying. Let's see. Dream research has traditionally relied on reports collected after someone wakes up, but people often wake with only spotty, distorted memories of what they dreamed. The dreamers can't say exactly what events occurred, and they certainly can't tailor their dreams to specific scientific studies. I feel like this is people will hold back when saying what happened in their dreams because they're oh, that didn't make any sense. But it's just of course it didn't. It was a dream. So when I talk about my dreams, I'll just say I'll be oh yeah, I ate a pizza and then I flew up in the air because I was oh wait, there's still pepperoni that I didn't get to eat or something. Because it's just that's the kind of wacky, batshit mentality you'll have in a dream. Or many times in a dream, I'll be someone will tell me something insane, and I'll be yeah, that makes sense. And then I wake up and I go, that didn't make any fucking sense whatsoever. Let's see, the special thing about lucid dreaming is that you can get even closer to dream content in a much more controlled and systematic fashion, says Martin Dressler, cognitive neuroscience at the Donders Institute in the Netherlands. Lucid dreamers who can perform assigned tasks and communicate with researchers during a dream open up tantalizing opportunities to study an otherwise untouchable realm. Oh my god, is this how they hook you up? Or is this just because... I assume the mask is maybe just because COVID. But even then, I feel like... Well, whatever. I feel like he could sleep without it, at least. So let's see, during Dream... they were like the astronauts of the dream world, serving as envoys to the mysterious inner spaces created by slumbering minds. They say Dream World and Dreamscape as if it's gonna be the same, like always, I guess. But it kind of just depends on that one person's mind. Let's see, so far, in tests in very small groups of lucid dreamers suggest that the strange realities we visit in sleep may be experienced more like the real world than imagined ones. With more emissaries enlisted. I love the vocabulary here, emissaries. Researchers hope to probe how sleeping brains construct their elaborate, often bizarre plots and set pieces. Besides satisfying age-old curiosity, this work may point to new ways to treat nightmares. Lucid dream studies could also offer clues about how dreams contribute to creativity, regulating emotions, or other cognitive jobs, helping solve the grand mystery of why we dream. It's a good, I think it's a good thing to study. But there's still a lot of problems to solve before lucid dream research can really take off. Chief among them is that very few dreamers can become lucid on demand in the lab. Those who can often struggle to do scientists' bidding or communicate with the waking world. Employing the best techniques to give more people more lucid dreams may assuage these issues, but even if it does, not all scientists agree on what lucid dreams can tell us about the far more common non-lucid kind. Trying to take you to the dreams way? That's true. What was I gonna say with oh yeah, something about the nightmares. Oh no, the falling asleep in the lab. Yeah, I think if I ever did a sleep study, I don't know if I could actually fall asleep in the lab. I think it would freak me out a little bit too much to have to be, you know, just watched by people or whatever. And I feel like it would take me a solid like hour just sitting in whatever bed they give me to try to fall asleep. Are lucid dreams real? I mean, lucid dreams are real, but I don't think what happens in the dreams are real. Let's see, tales of lucid dreams date back to antiquity. Aristotle may have been the first one to mention them in Western literature in his treatise on dreams. Often when one is asleep, he wrote, there is something in consciousness which declares that what then presents itself is but a dream. If Aristotle had lucid dreams often, though, he was probably an outlier. Only about half of people say they've ever had a lucid dream, while a mere 1% or so say they lucid dream multiple times a week. Yeah, not me. Modern enthusiasts use various techniques to boost their likelihood of lucid dreaming, such as reportedly telling themselves before bedtime that they will have a lucid dream, or make it a habit of checking whether they're awake several times a day, in the hopes that this routine carries over into their dreams, where self-check may help them realize they're asleep. But those practices don't guarantee lucidity. I also feel like if you make it a systematic thing, if you check every day several times, I feel like in the dream, like what's gonna happen, you know, say like, oh, I'm gonna fly, and then you try jumping to fly, and obviously in reality you don't. I feel like in the dream you'll try it and then you'll fall to the ground like normal, and then you'd be like, well, anyways, I'm gonna go to the bowling alley to have my date with like a gang member, you know, like with the mob boss or whatever. Just something like that. The rarity of lucid dreaming may be why modern science took some convincing that it's even real. For millennia, lucid dreamers' own testimonies were the only evidence that someone could be self-aware while catching Z's. and no scientist ever had lucid dreams, because they're constantly sleep deprived. And that's why they never truly believed it, because they're like, we didn't see any lucid dreams. Some scientists wonder if so-called lucid dreams were just brief waking hallucinations between bouts of sleep. Maybe. But within the last few decades, experiments have offered proof that lucid dreams are truly what they seem. It turns out, when someone in a dream purposely sweeps their gaze all the way left, then all the way right, their eyes can match those movements behind closed lids in real life. Oh. Does that mean that they aren't getting good REM sleep then? These motions, measured by electrodes near the eyes, stand out for the smaller optical jitters typical of REM sleep when most lucid dreams happen. This gives dreamers a crude way to signal they become lucid or send other messages to the outside world. Okay, so they're not like lucid dreaming and like talking. I guess they're using their eyes or whatever. Meanwhile, brainwaves and muscle paralysis throughout the rest of the body confirm that the dreamer is indeed asleep. Let's see, a person's eyes can smoothly track left and right movements when they are awake or in a lucid dream, but when someone closes their eyes and tries to imagine tracking that motion, their eyes pan in small jumps, suggesting that lucid dreams are experienced more like waking perception. So... Okay, so when you're awake and you try to move them left to right like that, lucid dreaming, like, there might be a few jumps. And I'm assuming this is saying if you're just fully asleep, not lucid dreaming, your eyes are very jumpy. Neuroscientists are just beginning to realize the potential of that line of communication. Lucid dream research has been enjoying a renaissance over the last decade, says neuroscientist Tor Nielsen. He directs the Dream and Nightmare Laboratory at the Center for Advanced Research in Sleep Medicine in Montreal. This renaissance has made it one of the cutting-edge areas of dream study. Ooh, this reminds me. Little Nightmares 3 was announced recently, and I am excited to play that whenever that comes out. One research team recently deployed experienced lucid dreamers to find out whether dream imagery is more like real-life visuals or imagined ones. While asleep, 6 lucid dreamers move their thumbs in either a circle or a line, or both, and trace that motion with their eyes. Participants repeat the same task while awake with their eyes open and their imaginations with their eyes closed. People's gazes pan jerkily when they track the imagined movements, as though they were viewing something low resolution. But in dreams, people's eyes track the movements smoothly just as in real life, the team reported in 2018 Nature Communications. It's been debated really all the way back to the ancient Greeks. Are dreams more like imagination, or is it more like perception, says study co-author Benjamin Baird, a cognitive psychologist and neuroscientist at the University of Texas at Austin. The smooth tracking data suggests that, at least in that sense, the imagery is more like perception. This and other early experiments offer a taste of what dreamstronauts could teach us, but any conclusions based on just a handful of dreamers have to be taken with a grain of salt. True. They're more like proof of concept studies, says Michelle Carr, a cognitive neuroscientist. It needs to be studying bigger samples. That means finding or creating more expert lucid dreamers. It's true, a lot of, especially, this happens a lot in social sciences, where we'll be like, oh, look, like this, like this one study or this, like, these 2 studies or whatever, and I'm like, I kind of want this to be done with, say, like, maybe hundreds of people rather than just, like, a case study. Because, of course, case studies can be helpful. But whenever someone points to a case study for, like, this is how things are, I'm like, I need a little bit more. You gotta give me that little bit more proof. What about REM? What about it? You're saying, like, are you talking about with the them moving their eyes around? Because it seems like even if they're lucid dreaming, they can still sort of, or they actually can control their eyes like a little bit better. But so far, they really haven't talked too much about REM sleep in here, which I'm also curious about. Strategies for lucid dreaming. We can probably, well, is there anything useful here? No, I know what REM means, but what about the rapid eye movement? Let's see, blah, 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 blah. I wouldn't be mucking around with my neurotransmitters every night, Baird says. Oh, I see. Investigations by Baird and others have shown that the supplement galantamine promotes lucid dreaming, probably by fiddling with neurotransmitters involved in REM sleep. But galantamine can be saddled with side effects such as nausea. And although lucidity itself does not appear to spoil sleep quality, yeah, I don't know if I would want to be taking something that would possibly give me nausea every night either. Here, let's see. Can we actually let this whole thing? Ooh, perfect. Let's see what we got here. Actually, what was this paper for? Would they study? Let's see, lucid dreaming. Nice. Okay, so I guess this is more just like the in-depth study that they did with everyone. See responses, dimensions of consciousness, questionnaire separated by lucid, non-lucid dreams, and glantamine dose. 0 milligrams, 4 milligrams, and 8 milligrams. Apparently sleep so you don't kick. Yeah, you will be paralyzed. I think that's why they're utilizing they did mention, didn't they mention something about fingers? My mind blanked on it. So they talked about making them look left to right, which they can, you know, I don't know. The question is does it happen each time, you know, when they try to look left to right? Let's see, repeat same task, eyes open. Okay, no, maybe I just brought that out of nowhere. Yeah, it looks like it's just their, like, signal to be like, hey, I'm lucid dreaming now. And then I'm guessing when they wake up, or they try to sort of, you know, there are a few things that they talked about here, as in just like, I guess, whether or not someone's lucid dreaming, which I think they just do with looking left to right with their eyes, versus like telling them about their lucid dream, which would definitely have to happen when they're awake. And like I said earlier, you know, it could be hazy and spotty. I think literally right. Yeah, spotty, distorted memories of what they dreamed. So the lucid dreamer would also have to hopefully have a pretty good memory of everything that went on. That's kind of like sleep paralysis, where your body is still paralyzed, but you're kind of awake. Lucid dreaming is just like you are dreaming, you are in the dream, and you can actually control, like, you are fully aware, like, hey, I'm dreaming. So like, now that I know I'm dreaming, I can imagine whatever I want to happen in this dream. Not quite daydreaming, but it's just like you have full control over your dreams, and you can, you know, control whatever happens in it. But for most people, usually once you hit that point of like, oh wait, I'm asleep, that's when their body, I guess, just like ejects them and is just like, no, you're awake now. That's why I was saying earlier, like, it's debated whether lucid dream was even real, because it was like, maybe this is just people like in a sort of half sleep state. But it seems like lucid dream is getting, I guess, a bit more ground in like proof. Hell, I guess even like this says, they're more like proof of concept studies. Let's see. Okay, so you can take glantamine. I'm curious. Well, let's see. Does this actually talk about glantamine much? Glantamine simulates lucid dreaming, double-blind, placebo-controlled crossover study. I just want to see if there's any information about the actual chemical itself. Also significantly increased dream recall, sensory vividness, and complexity. Interesting. Oh yeah, it did say it was a neurotransmitter. Integrated method, taking glantamine in the last third of the night with at least 30 minutes of sleep interruption with an appropriately focused mental set is one of the most effective methods for inducing lucid dreams available today. So I guess if you super want to do some lucid dreaming, shove some galantamine in ya and yeah. Okay, I guess this is the... I'll do that. Censor something out for you. Hold on, let me check that real quick. There might be an echo real quick. Oh, got it. So let's see, it looks like in REM sleep, REM sleep. I'm guessing this means like non-REM sleep. Import. Okay, so yeah, they wake him up, give him the galantamine, and try to get back to sleep in 30 minutes. Huh, okay. Interesting. Let's see, probably by feeling. Okay, so in 2020, what about Anthony Gailey as well? Some conscious aspects of business. What? Who is this? A hypnotist person? There probably is a lot of I would say crossover with trying to perform hypnosis and lucid dreaming type stuff. Again, like I was talking about earlier with like quote unquote reality shifting. I think like hypnotists will also use that sort of method of making someone fall asleep and then sort of subconsciously, you know, like telling them something so that when they wake up, they'll, I guess, believe it in their sort of relative delirium. Let's see, in 2020, Carr and colleagues reported that they'd coaxed 14 of 28 nappers to become lucid in the lab, including 3 people who had never before lucid dreamed. No drugs necessary. Oh, how'd they coax them? Before falling asleep, participants learned to associate a cue, such as a series of beeps, with self-awareness. Hearing the same sound again while sleeping reminded them to become lucid. Yeah, so it's this sort of weird state of like, you're asleep, but it's like, you don't really know how to make yourself aware, I guess, usually. Carr is particularly interested in finding out whether lucid dream can help people conquer nightmares, but researchers at Northwestern use a sensory coup strategy to get more lucid emissaries to carry out dream tasks for their experiments. It's pretty handy, and nice not having to use the galantamine. Let's see. 3 doses of galantamine, those took higher doses were more likely to have lucid dreams. Let's see, our method is kind of a shortcut. Northwestern cognitive neuroscientist Ken Poller. It doesn't require a lot of mental strain or the grueling sleep interruptions. Some other lucid dreaming techniques too. Yeah, basically get into that subconscious, and I guess this brings up a good point of like, there's probably not very good, it's probably not a very good standardized like way of inducing lucid dreaming. So you know, I'm sure some scientists would argue that like, oh no, like, you know, maybe like the galantamine messes with whatever, like, so it's not true lucid dreaming, and you know, so it's a matter of fighting with definitions to be like, okay, what exactly is lucid dreaming, like what consists of lucid dreaming versus just, you know, maybe just like being aware you're awake, but maybe not controlling it. Okay, I'll probably look up Anthony Gailey later. Let's see, another shortcut for researchers is to recruit dreamers from a special slice of the population, people with narcolepsy who are liable to fall asleep suddenly during the day. They're just champions at Lucid Dream, says Isabel Arnulf, a sleep neurologist who heads the sleep disorders clinic at man, I don't know how to pronounce that, but University Hospital in Paris. So it'd be T-Salpite or something. In 2018, Arnulf's team reported a study where 18 of 21 narcolepsy patients signaled lucidity during lab naps. Even with those impressive numbers, a couple of lucid nappers still couldn't control their dreams well enough to complete their assignment. Yeah, so like I said, you can be aware in a dream and still not really have control. Let's see, to do something in a dream that made them briefly stop breathing, such as swimming underwater or speaking. One said after waking that they'd simply forgotten to stop breathing while driving off a cliff, while another said they tried to speak but couldn't get any words out. Staying lucid and successfully wrangling dream scenarios present challenges for lucid dreamers, and scientists relying on them. On one say, lucid dreamers instructed to fill a dream room with objects such as a clock and a rubber snake ran into problems. The clock spun wildly, or the snake slithered away. That's a good point. In another experiment, lucid dreamers asked to practice throwing darts were waylaid by only having pencils to throw, or being pelted with darts by a nasty doll. Okay, I assume that was something specific. It's a lot harder than just passively lucid dreaming in your bed, says Mazurik, who has participated in several lucid dream studies at Northwestern. You realize, okay, I have to stabilize the dream. I have to remember what the task is. I have to do the task without the dream falling apart. Missions to the moon may be hard, but at least astronauts don't have to worry about forgetting who or where they are, or their spaceship suddenly turning into a banana. Despite these challenges, lucid dream expeditions are forging ahead and fast. In fact, an international crew of dreamfarers, including Zurich, recently embarked on their most ambitious mission yet, which is... when it comes to getting on the ground data, interviewing dreamers in real time is, well, the dream. Instead of just sitting back and watching dreamers do various activities, researchers could ask these agents about their experiences moment to moment, painting the realm of dreams in sharper detail than ever before. Reports of dream sensations, such as tasting certain foods, can be compared with those of actual sensations, Nielsen says. Similarly, one could test whether sexual pleasure, certain sounds, or other types of experiences are accurately simulated. These details, he says, might help probe the limits and mechanisms of dream production. Karen Cancoli is especially excited about giving people assignments mid-dream. I don't know, I feel like that would I feel like you should start with having a goal in mind going into the dream, because you tell someone something mid-dream and then it's just gonna fuck it up. Say researchers want to know how much dreams help with creative problem solving. If dreamers are assigned a problem before sleep, they're liable to mull it over as they nod off. Even if it feels like the lucid dream, maybe it's really the time as you're falling asleep that helped you solve the problem, says Cancoli. Okay, fair. A cognitive neuroscientist. Airdropping a puzzle straight into a dream could better isolate the usefulness of that specific part of sleep. Depending on how complex the puzzle is, I wonder how you quote-unquote airdrop it in. Let's see, there's a whole medley of theories about why people dream. You give them the trolley problem while they're sleeping, and then they have an existential crisis as they're asleep. There's a whole medley of theories about why people dream, from honing skills to tapping into creativity to processing memories or emotions. But if you can't control the dream in real time and then study the outcome, then you never know if the dream is really doing anything, Cancoli says. So a few years ago, she, Arnulf, Dressler, and others decided to find out if people can receive and respond to outside input while dreaming. I definitely know there have been times where I've been asleep when like the TV's on or something, and I'm fully, like, I realize in hindsight, like, I was fully listening to the TV, and that was affecting my dream and what was happening in it. But only in hindsight. Like, obviously when I was sleeping, I was just doing whatever. So, 36 people took snoozes at Northwestern Arnulf's lab, Dressler's lab, or another lab that was in Germany. Once sleepers signaled that they were lucid, researchers spoke yes or no questions or math problems in the sleepers' ears. Or for the Germans, lights flashing different colors conveyed math questions in Morse code. Before conking out, dreamers were told to answer whatever questions they received with eye signals or by smiling and frowning.
Facial muscles are less inhibited than other muscles during REM sleep, Arnulf explains. Okay. Someone smiling in a dream may not make that expression in real life, but electrodes on the face can register tiny corresponding muscle twitches. I see. Out of 158 attempts to interrogate lucid dreamers, 29 total correct responses came from 6 different people. Okay, so I assume it would be like easy, a real easy thing to confirm that they're even giving real responses versus just their eyes moving. Those 6 range from newbie to frequent lucid dreamers, including Masaryk, who heard scientists' questions while dreaming he was in the Legend of Zelda game. The rest of the attempts yielded 5 wrong answers, 20 ambiguous ones, and 96 non-responses. Ooh, okay. Yeah, so 29 total correct responses is cool and all, but there's a whole lot of ambiguous and non-responses. When Concoli first saw someone correctly answer a question in their sleep, my first reaction was to not believe it. But for 26 of those 29 correct responses, a panel of independent sleep experts unanimously agreed that the dreamers were in the throes of REM sleep when they replied. Nearly 400 attempts to reach sleepers who hadn't signaled lucidity netted a single correct response, bolstering the researchers' confidence that correct answers from lucid dreamers weren't flukes. The results appeared in 2021 Current Biology. Send yourself to sleep. Then we'll experiment and we'll give you some answers and problems to solve. just move your eyes left to right, left for yes and right for no. So your interesting questions in your dream. Graph below, Mazurik's eye motions that indicate he is lucid appear as 3 big up-down sweeps. Eye signals answering 2 to researcher simple math questions appear as 2 big up-down sweeps. Oh, okay. I'm lucid. Oh, damn, this is in like half a minute? I thought they would like give it a bit more time. Was 8 minus 6, 2 was 8 minus 6, 2. I'm also curious though, like, what's the maximum amount of, I guess, eye movements that you can make before it starts to say like trail off, If you said like, oh, give me, like 20 minus 10, like, would they actually be able to accomplish 10 eye sweeps or whatever? I was astonished, says Robert Sitgold, a cognitive neuroscientist at Harvard Medical School who studies dreams but not lucid ones. I had no question but that these people are in fact listening and are in fact having lucid dreams at the time of the communication. And that opens up all sorts of possibilities. Arnulf and others have since asked lucid dreamers to smile or frown as their dreams become more or less pleasant, with the goal of understanding how dreamers experience emotion. Another study, not yet published, tracked when lucid dreamers answered or ignored research questions to see how people tuned in and out of the real world while dreaming. Knowing which signals break the dream reality barrier could help uncover the mechanism of the brain's disconnection from the external world, which is huge, Baird says. It could even be relevant for other states of unconsciousness, he adds, such as when someone is put under for surgery. Limits lucidity. Here we go. Even if researchers get all the expert lucid dreamers they need to run all their desired experiments, there's still one major sticking point to this whole field of study. The biggest issue is how far can you push these results to dreaming in general, Sitgold says. Imagine, for instance, that lucid dreamers get better at a skill by practicing in their dreams. It's not clear that people who just happen to have normal dreams about doing those activities without self-awareness would reap the same rewards. It's a little bit like recruiting Major League Baseball players to give you some baseline data on how far people can throw balls, Sitgold says. Existing data do suggest that lucid dreamers may have access to parts of the brain that normal dreamers don't. I always hate the phrasing of like, don't have access to parts of the brain or something, or like anything that tells you like, oh, we only use like 10% of our brain or whatever. It's like, no, that's not true. It's like we use all of our brain. We don't use a percentage of it. The lone case study comparing fMRIs of someone's lucid and non-lucid REM sleep hints that brain areas linked with self-reflection and working memory are more active during lucidity. Those data come from just one person, and it's not yet clear how such differences in brain activity would affect the outcomes of lucid dream experiments. What else? There's another... I mean, they did say that they had some people who had little to no dreaming or lucid dreaming experience before. So I mean, I feel like that's pretty good too. Let's see, brain clues to lucid dreams. Functional MRI scans. Alright, let's see, maybe active during lucid dreams and during normal sleep. I will believe, however, like, maybe a part of the brain not being used as much during normal sleep, but being used, like, when they're awake. So that I can kind of see. So let's see, lateral parietal cortex is involved in working memory. Dorsolateral prefrontal cortex and frontopolar cortex are involved in working memory and introspection. Activity in neurotemporal cortex may make lucid dreams brighter and more detailed than normal dreams. That's another thing. Having to tackle how people's imaginations are wildly different as far as what they can actually imagine. Some people can imagine, like, you know, full bright colors and whatever, and other people might only think and say, like, words or something. So that can also, I feel like, have a pretty big difference in this. Some researchers, including Dressler, I'm gonna take a water break real quick. Including Dressler resist the idea that lucid dreams are profoundly different from non-lucid ones. Lucid dreaming is not a strict all-or-nothing phenomenon, he says, with people often fluttering in and out of awareness. That suggests that lucid and non-lucid dreaming are in principle something very similar on the neural level, and not two completely different animals. Perhaps lucidity affects some aspects of the dream experience, but not all of them, Baird says. In terms of how dreams look, he says, it would be very, very surprising if it was somehow completely different when you become lucid. Alright. A more thorough inventory of the differences in brain activity between lucid and non-lucid dreams might help settle those questions. But even if lucid dreams don't represent dreams in general, Nielsen still thinks they're worth studying. This is a type of consciousness that has intrigued and amused people for centuries, he says. It would be important for science to understand how and why humans have this extraordinary capacity for intentional world simulation. Huh, well, we'll see how, I would have to look into, I guess, we've had lucid dreaming, I feel like, like I said, since, like, Aristotle. But if we're more readily studying it or having better studies with it, so we'll see how it is in another decade or so. Alright, so this next one. Unlocking the Iceman. Advanced genetic analysis of Otzi reveals surprising ancestral roots and appearance. Let's see. Perfect. Alright. So this one I have no clue what it's about. I just pulled up and I was like, oh, what would be surprising about ancestral roots and appearance? So let's see, also known as the Iceman, as genome resequence. With advanced technology, actually, you know what? Well, I'm sure I'll say it. So I was gonna say, I don't know too much about the Iceman. Revealing they had shared more genes with early Anatolian farmers than other Europeans of his time. Contrary to previous beliefs, Utsi had advanced hair loss, possibly being bald, had a notable darker skin tone than was earlier thought. Furthermore, while his genes indicated predispositions to obesity and diabetes, his active lifestyle likely counteracted these genetic tendencies. Okay, I'm gonna sneeze. Okay. Utsi's genome was decoded for the first time more than 10 years ago, which was also the first time the genome of a mummy had been sequenced. This sequencing offered pivotal insights into the genetic composition of prehistoric Europeans. Recent advances in sequencing technology have now enabled a research team from the Max Planck Institute for Evolutionary Anthropology and URAC Research to reconstruct Utsi's genome more accurately. Results of this recent analysis refined the Iceman's genetic picture. Compared to other contemporary Europeans, Utsi's genome has an unusually high proportion of genes in common with those of early farmers from Anatolia. Additionally, contrary to previous findings, at the time of his death, Utsi had advanced hair loss and may have even been bald. God, look at that just like disgusting clump of his crotch. Moreover, his skin was darker than previously thought. Woodsy's genes also show a predisposition to diabetes and obesity. Huh. Let's see, since 2012, which is when Woodsy's genome was sequenced for the first time, DNA sequencing technologies have advanced enormously. This new study reveals that compared to other contemporary Europeans, Woodsy's genome had an unusually high proportion of genes in common with those of early farmers. You are throwing me the same paragraph over and over. European genetic lineage. Present-day Europeans predominantly exhibit a genetic composition that stems from the admixture of three ancestral groups. Western hunter-gatherers gradually merged with early farmers who migrated from Anatolia about 8,000 years ago, and who were later on joined by steppe herders from Eastern Europe approximately 4,900 years ago. In the initial analysis of the Iceman's genome, genetic traces of these steppe herders was unveiled. However, the refined new results no longer support this finding. The reason for the inaccuracy is that the original sample had been contaminated with modern DNA. Whoa. Since that first study, not only have sequencing technologies advanced enormously, but many more genomes of other prehistoric Europeans have been fully decoded, often from skeletal finds. This has made it possible to compare Otzi's genetic code with his contemporaries. Damn, so I guess just someone's, like, skin cells or something could have fallen into a tube. The result is that among the hundreds of early European people who lived at the same time as Otzi and whose genomes are now available, Otzi's genome has more ancestry in common with early Anatolian farmers than any of his European counterparts. I wonder how he's been, preserved, you know? I guess maybe... like, it looks like he's got a bed, or maybe this is a table or something. So I suppose they could throw him in a freezer. But I wonder if they would want to do something, say, like liquid nitrogen or something. Yeah, he's definitely almost giving us a solid dab, but he doesn't quite understand it yet. Otzi was truly ahead of his time. Alright, so let's see. Okay. Okay, ancestry and appearance. Research team posits that the Iceman came from a relatively isolated population that had very little contact with other European groups. We're very surprised to find no traces of European or Eastern European steppe herders in the most recent analysis of the Iceman genome. The proportion of hunter-gatherer genes in Otzi's genome is also very low. Genetically, his ancestors seem to have arrived directly from Anatolia without mixing with hunter-gatherer groups, explains Johannes Kraus, head of the Department of Archaeogenetics at the Max Planck Institute for Evolutionary Anthropology in Leipzig, and co-authored the study. The study also yielded new results about Otzi's appearance. His skin type, already determined in the first genome analysis to be Mediterranean European, was even darker than previously thought. It's the darkest skin tone that's been recorded in contemporary European individuals, explains anthropologist Albert Zink. It was previously thought that the mummy's skin had darkened during its preservation in the ice, but presumably what we see now is actually largely Otzi's original skin color. Knowing this, of course, is also important for the proper conservation of the mummy. Our previous image of Otzi is also incorrect regarding his hair. As a mature man, he most likely no longer had long, thick hair on his head, but at most a sparse crown of hair. His genes, in fact, show a predisposition to baldness. Damn. I like to imagine that they imagine Otzi with, like, long, flowing locks, just like luscious, beautiful hair. And they're just like, nah, he was balding. Like, he might not have had any hair when he died. Alright, so let's see. This is a relatively clear result and could also explain why almost no hair was found in the mummy, says Zink. Genes presenting an increased risk of obesity and type 2 diabetes were also found in Otzi's genome. However, these factors probably did not come into play thanks to his healthy lifestyle. Healthy, quote unquote. Yeah, I mean, I'm sure there could be other things, but I just see that they brought up the risk of obesity and type 2 diabetes. Huh. Alright, well sweet. That was a quick one. We've also got researchers develop method for upcycling plastic waste into soap. So basically, they found a way to use some plastic as soap. So that's pretty good. Team led by Virginia Tech researchers has developed a new method for upcycling plastics into high value chemicals known as surfactants, which are used to create soap, detergent, and more. Plastics and soaps tend to have little in common when it comes to texture, appearance, and most importantly, how they're used, but there's a surprising connection between the two on a molecular level. The chemical structure of polyethylene, one of the most commonly used plastics in the world today, is strikingly similar to that of a fatty acid, which is used as a chemical precursor to soap. Both materials are made of long carbon chains, but fatty acids have an extra group of atoms at the end of the chain. Gu Lian Greg Liu, associate professor of chemistry in the Virginia Tech College of Science, had long felt the similarity implied that it should be possible to convert polyethylene into fatty acids, and with a few additional steps to the process to produce soap. It's pretty good. The challenge is how to break a long polyethylene chain into many short but not too short chains, and how to do it efficiently. Liu believed there was a potential for a new upcycling method that could take low-value plastic waste and turn it into a high-value, useful commodity. Having considered the question for some time, Liu was struck by inspiration while enjoying a winter evening by a fireplace. I was just sitting there by my fireplace. It was a cold, cold winter day. I watched the smoke rise from the fire and thought about how the smoke is made up of tiny particles produced during the wood's combustion. Although plastic should never be burned in a fireplace for safety and environment reasons, Liu began to wonder what would happen if polyethylene could be burned in a safe laboratory setting. Be good, I guess. Would the incomplete combustion of polyethylene produce smoke just like burning wood does? If someone were to capture that smoke, what would it be made of? Interesting. Firewood is made of mostly polymers such as cellulose. Combustion of firewood breaks these polymers into short chains and into small gaseous molecules before full oxidation to carbon dioxide, said Leo. Holder of the... Okay. If we similarly... I love how many times in an article we'll mention like the same person's name and then give them some other descriptor or just like where else they work or saying where they work like 10 times. If we similarly break down the synthetic polyethylene molecules but stop the process before they break all the way down to small gaseous molecules, then we should obtain short-chain polyethylene-like molecules. So go on, throw your plastic into a fire and collect the molecules that come out of it. I do like that way of thinking. I mean, I thought he was gonna say even like he was watching TV or something, but it was just like imagine this man staring into a fire and then being like, I like to imagine him being like, God, I wish we could get rid of all this fucking plastic. And he's like, but what if? What if? So with the help of Xin Shu and Eric Muyanezi, two PhD chemistry students in Liu's lab, Liu built a small oven-like reactor where they could heat polyethylene in a process called temperature gradient thermolysis. At the bottom, the oven is at a high enough temperature to break the polymer chains, and at the top, the oven is cooled to a low enough temperature to stop any further breakdown. After thermolysis, they gathered the residue, similar to clean soot from a chimney, and found that Liu's hunch had been right. It was composed of short-chain polyethylene, or more precisely, waxes. Interesting. This is the first step in developing a method for upcycling plastics into soap, Liu said. Upon adding a few more steps, including saponification, I almost read this wrong as soapification, the team made the world's first soap out of plastics. To continue the process, the team enlisted the help of experts in computational modeling, economic analysis, and more. Some of these experts were introduced to the team through connections with the Macromolecules Innovation Institute at Virginia Tech. Together, the group documented and refined the upcycling process until it was ready to be shared with the scientific community. Hold on, I think my dog wants out. But she is a little dumb, but I love her because there are two doors to this room and the other one is very clearly open. She's trying to go out the first door. Alright, there we go. She, after taking her to it, she was made aware that the other door was open. Very clearly in front of her. And now she has come back in the room to stare at the first door. Okay. You just stay there then. Yeah, this is huge for soap. I did see one little thing where it was saying, like, you know, they were like, we're not going to be able to do this with, like, all plastic. But it's a good, like, first step for sure. Where was I? So, new route for plastic upcycling without using novel catalysts or complex procedures. In this work, we have shown the potential for a tandem strategy for plastic recycling, said Chu, lead author on the paper. This will enlighten people to develop more creative designs to upcycling procedures in the future. And for anyone that doesn't know, I hate to break this news to you, but a lot of plastic recycling is actually bullshit. Like, plastic doesn't actually get recycled all that much, if at all. It is actually a pain in the ass process to recycle. But this, like, you know, haven't thrown this on the chart, it's like, oh shit, it's like, that's actually good. Like, that does mean we can have a pretty good reason to recycle plastic. Or at least use it for something. Although polyethylene was the plastic that inspired this project, the upcycling method can also work on another type of plastic known as polypropylene. These two materials make up much of the plastic consumers encounter every day, from product packaging to food containers to fabrics. One of the exciting features of Liu's new upcycling method is that it can be used on both these plastics at once, meaning that it's not necessarily separate the two from each other. This is a major advantage over some recycling methods used today, which require careful sorting of plastics to avoid contamination. The sorting process can be quite difficult because of how similar the two plastics are to each other. Another benefit of the upcycling technique is that it has very simple requirements: plastic and heat. Although the later steps in the process require some additional ingredients to convert the wax molecules into fatty acids in soap, the initial transformation of the plastic is a straightforward reaction. This contributes to the method's cost effectiveness as well as its comparatively small environmental impact. Man, again, I love... Here, let me see if I can pull some up. So, it talked about how, like, cellulose, like, imagine, yeah, this repeated, like, over and over and over again. And that's essentially what you're burning in a fire with wood. And then just being like, polyethylene is a, like, you know, big chain molecule. Like, what if we just burn that shit up too? I mean, hey, I'm glad it worked. You know, like, sure, whatever, fuck it. But I love that it was just that very simple idea. Another benefit of the upcycling technique is that it has very simple... Okay, never mind. For upcycling to be effective on large scale, the final product must be valuable enough to cover the cost of the process and make it more economically attractive than alternative recycling options. I'm sure depending on which company you talk to, they'd be like, oh, fuck yeah, we'll sell this soap and we'll make a shitload of money due to it being green. Assuming that it doesn't give off any kind of bad gases, because if it starts being made in, like, a much wider scale, say like a factory or something, I don't think we want all that burning plastic, you know, like huge amounts of burning plastic residue or whatever. Eat an oil bath. Okay, that's with the waxes. I'd say, although soaps may not initially seem like a particularly expensive commodity, they can actually be worth double or triple the price of plastics when compared by weight. Oh yeah, that's another thing. Since a lot of plastics recycling, since it's more expensive to recycle, companies will just be like, we're not gonna recycle it then, we don't give a fuck. So it's like, man, that's sad. Okay. It was fun that last time we played, and someone got into the core or whatever, and started tearing the entire ship apart, or station apart. Let's see, currently the average price of soap and detergent amounts to about $3,550 per metric ton, and that of polyethylene is about $1,150 per metric ton. Furthermore, the demand for soaps and related products is comparable to the demand for plastics. This research lays the groundwork for a new way to reduce waste by channeling used plastics into the production of other useful materials, Lou said. Over time, he hopes recycling facilities around the world will begin to implement this technique. If so, then consumers can expect to one day have the opportunity to buy revolutionary, sustainable soap products that also lead to reduced plastic waste in landfills. Yeah, that was definitely a good, like, I don't even know what, like, comedy bit to call it exactly, but just saying hey to a chainsaw killer and then walking past him and then be like, wait a second, that was the dude. It's as if he had a mustache on and we just fell for it. Like, he threw on some glasses and we really got Clark Kented. For this reason, turning plastics into soaps can be demonstrated to be economically viable, had Liu, who is also an affiliated faculty member of the nanoscience program. Part of the college. See? My god. like, just telling me this whole man's resume. Should be realized that plastic pollution is a global challenge rather than a problem of a few mainstream countries. Compared to a sophisticated process and complex catalyst or reagent, a simple process may be more accessible to many other countries worldwide, Xu said. I hope this will be a good start for the war fighting plastic pollution. I pulled up another version of this article earlier, and it had these like very bright colored soaps on it. But I'm thinking, oh, this might not be the actual soaps that they had. Because there's one point I quickly read in the article where it was like, you know, it was like, oh, like the bright colors will be attractive to people. I'm just like, yeah, I think anyone, like, I think even the bright colored soaps that we have now, people like a lot. All right, one more to go through. And I think this one will be kind of short, so it might be done soon. The scientists are intentionally bleaching and cryopreserving coral. Which I was kind of thinking, I'm like, weren't corals kind of come back? I'm surprised that we're bleaching some of it. So let's see, preserving coral reef. Oh yeah, I didn't even change from the Iceman thing. Oops. That's fine. Not like anyone's searching for these cells in particular. Preserving coral reef ecosystems is absolutely vital to ensure a stable, healthy ocean environment. Sadly, ongoing climate crises continue wreaking havoc on experts' efforts to avert the worst effects of die-offs and coral bleaching. With new advancements, however, one potential solution could involve cryopreservation, collecting containing coral samples at temperatures as low as negative 321 degrees Fahrenheit. According to a recent study in Nature Communications, a team at Texas A&M University has developed a novel cryopreservation technique called isochoric vitrification. First, fragments of coral are actually strategically bleached in a lab using a combination of menthol and light. And coral fragments temperatures are quickly lowered to negative 196 degrees Celsius or nearly negative 321 degrees Fahrenheit inside special aluminum containers. But despite the low temperatures, the coral is cooled without immediate injury. Because it's any kind of cryopreservation or like nitrogen-based freezing, it's like extremely quick, so hopefully reduces any kind of damage that regular freezing would have with ice crystals and such. Trying to remember what else. Oh yeah, I'm curious why the bleaching, what that accomplishes before they cryopreserve it. Although any kind of bleaching often removes symbiotic algae crucial to coral photosynthesis, not doing so ahead of isochoric vitrification results in the formation of deadly ice pockets. Okay, so that's specifically to get rid of ice pockets. for the cryopreservation. By removing the algae ahead of vitrification, however, fragments could be preserved in a glassy state via submerging the coral in the chemical solution in aluminum containers cooled using liquid nitrogen. Okay, so it is liquid nitrogen. A layer of the coral could be revived by slowly warming samples with the reintroduction of filtered seawater. Yeah, because the, like any kind of ice crystals or ice pockets that are created when doing regular freezing, that can mess with the molecular structure of things, especially as you're unfreezing it. So it's good to reduce that if you're doing something, if you're freezing something for preservation. Let's see, but don't go doing it with like your food or something. You can have a little bit of ice pockets as a treat. You'll be fine with it. So let's see, is this collaborative marriage of fundamental thermodynamic advancements, fundamental advancements in coral biology and husbandry that have enabled our breakthrough success in whole coral cryopreservation? Matthew Palm explains in a statement. Although the team notes that coral cryopreservation has already been used in the past, the methods require collecting samples during coral reproduction cycles. Such breeding periods only occur a few days in a year, often in difficult to reach areas. In contrast, isochloric vitrification allows researchers to harvest and preserve coral regardless of time of year. What's more, the new method is vastly simpler than alternative cryopreservation techniques. Yeah, the music is very happy. This isn't a playlist like I put together. It was just one I found randomly. So that's why it's pretty varying in all of its music. But yeah, this is pretty good. Let's see, compared to other emergent vitrification techniques which frequently require lasers, electromagnetic implements, or other high-tech laboratory equipment, our isochloric vitrification approach requires no moving parts or electronics, and the protocol can be implemented by a field technician with no background thermodynamics. Cool. This is essential to the practicality of any conservation technique, because when this is deployed in real marine field stations, the high-tech lab infrastructure common to many laboratories will not be available. Okay, I read marine for like, for water, and at first I thought it meant marine as in like military. And I was like, what the hell? Yeah, science music playlist, to be honest, I've looked up quote unquote science music, and a lot of it will be like either copyright music, and I guess I'm not too against it, but just like a lot of things will come up be like music for programming, and I'm like, I don't know. For me, I'm like, that's not science. I want one that says science in it. I think that was the one that I pulled up last time. But I also like changing it up, even though I don't recognize any of the songs. Alright, let's see. From a purely technological perspective, this technique is simple, rugged, and ready for the field, Palm explained via the announcement. After honing their new technique, researchers tested the isochloric vitrification process in coral fragments at the Hawaii Institute of Marine Biology. Currently, coral samples post-thaw lifespans post-thaw only lasted less than 24 hours, but the team believes reducing the procedure's overall stress effects will extend the method's viability. Coral reefs are essential to the baseline health of our oceans, and cryoconservation of endangered coral species can help to ensure that these invaluable and marvelous organisms do not go extinct. Palm. All right, and that's all the news I had pulled up for today. But you know what, Ranga, you're right. I am curious what I could get from Mad Scientist music. Let me take a quick look-see before we end. What the hell is this? Mad Scientist Barbecue. Oh, it's a YouTube channel. Oh, fuck. I can't look at this. That's gonna make me hungry as hell. Oh, God. Muzak. Spooky music. The Alchemist Tower. Let's see. Okay. Yeah, this definitely does have that fantasy feel of like some weird magic shit's going on. Almost Harry Potter music. But to be honest, wouldn't be against it. Hold on. At the end of this playlist is a 10-hour video. Halloween white noise, mad scientist laboratory sounds. I hope there better be cackling in the background. Okay, hold on. I'm looking at the, like, you know, the most played parts in this video. There's one part in particular that has a huge jump. Alright, let's see what this is. I don't know if it's gonna be loud or something. Once it loads. Perfect for Halloween parties. We need some wolves howling, graveyard bells, and the occasional zombie heard in the wind. It's true. This is literally, someone said, like, this is just a, like, this is basically a jacuzzi. That's basically all this is. Okay, what's the, hold on, I guess I went past the thing. I just want to see what the one thing is. There has to be a timestamp, right? Usually whenever there's a big jump like that, someone has a timestamp in the comments. Someone just commented, "All boy dark force." Sure, bud. Alright, I think it's right here, maybe? Let's see. I don't understand. Skipping through it. Alright, well, whatever. Yeah, that'll be the end of today's science news stream. Pretty good stuff. But I think I'll end for today. Man, I realized that these kill my voices a lot more than everything else. Alright. And then we'll do some, let's see. We're gonna go ahead and do Mario tomorrow, then we'll continue all the other things that we're playing for the rest of the week. And I'll be gone during the weekend. But, thank you to everybody that came by today. I appreciate ya. Hope you had a good time and learned something today. Hope everyone has a good rest of their day or night. And I will see you next time. And remember, as the one article we read, to burn your plastics. But, like, not burn them too much, but, like, melt them enough so that you can collect the waxes and create soap. Goodbye, everyone.